In this last portion of the lecture, we will review the process of translation. Remember that translation involves amino acids being joined together with a type of covalent bond called a peptide bond to create a polypeptide. Proteins are folded polypeptides. The amino acids are loaded onto tRNAs in the cytoplasm. It's the tRNAs that deliver the correct amino acid to the ribosome. The correct tRNA is the one that contains the anticodon that will match the codon exposed on the mRNA molecule. It's the ribosome that exposes one codon at a time on the mRNA. Only the tRNA with the correct anticodon will be able to come in and interact with the codon. When that interaction occurs, the amino acid that is on the tRNA will be removed and added onto the growing strand of amino acids. In the drawing on the right-hand side of the slide, you can see this process. The black strand represents the mRNA molecule, and the little colored teeth that are extending from it represent individual bases. The gray molecule represents the ribosome, both the small subunit and the large subunit. These twisted and turned black molecules represent the tRNAs, and you can see that they too have bases jutting off of them. This is the anticodon, a group of 3 bases on the tRNA that are complementary to certain combinations of bases on the mRNA. Each tRNA carries a different amino acid. The ribosome is going to bind to the mRNA at the start codon. The only tRNA that can come in and interact with the codon is the tRNA that's carrying the amino acid that matches the start codon, and that amino acid is methionine. The methionine will be placed, the ribosome will travel to the next codon in a process called translocation. That new codon will be exposed, and a new tRNA will come in, carrying a new amino acid. The ribosome will continue to translocate down the length of the mRNA until the entire mRNA has been translated and a new polypeptide has been produced. Remember that ribosomes are found in all living cells, whether they are prokaryotic or eukaryotic. It's the ribosome that is the machinery of translation. Ribosomes are made out of 2 subunits, a small subunit and a large subunit. Both of these subunits contain proteins and ribosomal rRNA. Each subunit contains both protein and rRNA. It's the job of the small subunit to hold the mRNA in place and expose one codon at a time. The large subunit has the job of building peptide bonds between incoming amino acids. Remember, if you were to slice the large subunit of the ribosome in half, you would find empty tunnels inside of that molecule. Those tunnels, or sites as they're called, are involved in the step-by-step process of taking the amino acid off of an incoming tRNA, adding it to the growing chain of amino acids, and then releasing that tRNA molecule so it can once again be loaded up with a new amino acid. During translation, all three of those sites inside the large subunit will be occupied at the same time. As a new tRNA enters, it comes into the A site. What was in the A site will move over into the next P site. What was in the P site will shift over into the E site. Every time the ribosome translocates, whatever is located in the three sites shifts one position. So the sequence of events involves a tRNA coming in and binding anticodon to codon at the A site. Once it's in the P site, the amino acid will be removed from that tRNA and added to the C terminus of the growing polypeptide chain. Once it's in the E site, the now empty tRNA will be released into the cytoplasm. Every time the ribosome translocates, everything in the three sites shifts over one position so that a new tRNA can come into the A site. Translocation occurs about 20 times a second in prokaryotic cells. It's slower in eukaryotic cells, occurring about twice every second. On this slide, we can see the process of translation occurring. Again, this artist has sliced the ribosome right down the middle so we can look inside. This part of the golden yellow structure represents the small subunit of the ribosome. Remember, the job of the small subunit is to hold the messenger RNA in place and to align one codon at a time. This is the large subunit, and inside of it you can see three distinct channels or sites labeled A, P, and E. Notice that there is another tunnel at the top of the large subunit of the ribosome that is used to hold on to the growing chain of amino acids, the polypeptide. Now in this drawing, we are in the midst of translation. All three sites in the large subunit are occupied. A brand new tRNA has just entered the A site and interacted with the exposed codon. You'll notice that the codon reads GCU, so the anticodon will be complementary to that, CGA. Only this tRNA will have the correct anticodon that can interact with this codon, so only this tRNA will be allowed to come into the A site. This tRNA carries the amino acid that is coded for by GCU. And if we were to go to the genetic code table, we would find that GCU is the codon for alanine. So the tRNA molecule is really the middleman of translation. The tRNA is doing the job of matching up the information or the code in the codon with the correct amino acid for that particular code. Now the ribosome is going to translocate and shift over to the next codon. What's in the A site is going to shift into the P site. And it's at the P site that the alanine will be joined with a covalent peptide bond to the amino acid sitting next to it, which happens to be serine. The amino acid will be taken off of the tRNA in the P site and added on to the growing chain of amino acids. The ribosome will then translocate to the next codon, and what's in the P site will be shifted over to the E site. This is now going to be an empty tRNA. It's already given up its amino acid, and it's now ready to exit the ribosome and enter the cytoplasm so that it can once again be loaded up with its amino acid. tRNA molecules get used again and again and again. Remember that both subunits of the ribosome contain protein and rRNA. But it's interesting to note that the active site in the large subunit, the part of the ribosome that is capable of building a peptide bond, consists entirely of rRNA. The active site of the ribosome is catalytic RNA. During the elongation stage of translation, as the ribosome travels down the length of the mRNA and new amino acids are brought in according to the code in the mRNA, and new peptide bonds are built, that process requires energy. It's interesting to note that this energy is not supplied by the molecule that you would think. It's not supplied by ATP, but rather the similar molecule called GTP. Once elongation is underway, additional ribosomes are going to come on and bind with the mRNA. In other words, it's important to remember that an mRNA does not get translated once. An mRNA is going to get translated several times before it's degraded. So several copies of that protein will be made. This creates a structure that's referred to as a polyribosome. In other words, several ribosomes working along the mRNA, creating a long polypeptide. That's what you can see in the images on the right side of this slide. Here's the drawing, and here's an actual electron micrograph. In the drawing, the mRNA is represented by a yellow strand. This is the 5' end, this is the 3' end. The ribosome will attach over near the 5' end of the mRNA at the start codon. Each ribosome is represented by a yellow circle. The green that you see emerging from the ribosome is the growing polypeptide, the string of amino acids. So as a ribosome travels down the mRNA, that string of amino acids gets longer and longer and longer. Notice that there is not one ribosome traveling down this mRNA, but many of them. Now, if you look at the electron micrograph, you can see what it actually looks like in a living cell. Many ribosomes attached to one mRNA, each ribosome producing a long chain of amino acids according to the code in the mRNA. Now, just as we talked about for transcription, there is a specific series of events that will terminate translation. Again, we're going to use bacteria as our model organism. In this drawing, you can see 3 stages across time. If we start on the left, you can see that there will come a point where the ribosome is going to translocate and land on a stop codon. Recall that in the genetic code table, there is only one start codon, but there are 3 different stop codons. Any time that a ribosome encounters a stop codon, translation is going to stop. Now the ribosome can't tell the difference between a stop codon and any other codon that it encounters. So how does translation stop? The stop codon is a codon. It has 3 bases in it. And there will be a tRNA that has the correct anticodon. That tRNA is going to enter in and bind anticodon to codon, just like any other tRNA. But here's the difference. The tRNA that matches a stop codon is referred to as a release factor. And it gets that name because it, unlike all the other tRNAs, does not carry an amino acid. So all a release factor is a tRNA that does not carry an amino acid. When a stop codon is encountered on the mRNA, the release factor will enter the A site, and when the ribosome translocates, the release factor will enter into the P site and have no amino acid to offer for the growing strand, the growing polypeptide. That's going to cause translation to be disrupted. The polypeptide is going to be released. The other tRNAs are going to leave their respective sites, and the small and large subunit will disengage from the mRNA. It's tempting to think that the end of translation is the end of the process of building a protein, but of course that's not true. What's produced during translation is a polypeptide, a long chain of amino acids. Polypeptides are large. They can contain hundreds or even thousands of amino acids, depending on the gene that they are produced from. That polypeptide needs to be folded into a protein in order to be functional. You can imagine that with hundreds or thousands of amino acids involved, and each amino acid having its own R group, a strand of amino acids could be folded up in many different ways. But there is only one correct fold for each protein. That means that the folding process has to be guided. In other words, proteins are not allowed to simply spontaneously fold most of the time. Instead, the folding process is guided by molecules called molecular chaperones. Other modifications have to be made to proteins as well. Many proteins are going to have portions removed. Some proteins are going to have carbohydrate added or lipid added. In a eukaryotic cell, the protein is going to travel through the organelles that we have discussed, the rough endoplasmic reticulum, the Golgi apparatus, and the transport vesicles that are going to take them to their ultimate destination. So, translation is not the end of protein production, but merely the beginning. We will be discussing protein processing and protein trafficking in a future lecture. So stay tuned.